Hi, good morning people of the world, Francis here. I thought today I would go over the coronavirus gene that's been mapped now, mapped in very good detail for the COVID-19, which has been causing all the anarchy over the last 18 months, a year and a half or so. Single strand RNA, it's got 30,000 nucleotides, okay? This is the actual RNA. So at one end is the 5' untranslated region. That is a section that is basically, it doesn't have any genes and it makes no proteins. But that does interact, it forms molecular interactions with other, with viral and host cell proteins, human cell proteins, okay? There's two genes here called open reading frame 1A and 1B, open reading frames. These encode 16 non-structural proteins, okay? Then there's a gene for the spike protein, which we all heard about in the news, the thing that sticks to cells. And then there's two other genes which are involved with sort of structural or accessory proteins. And then there's this E is actually it's a membrane protein. It's a very small membrane protein that is used for virus assembly plus the membrane permeability of the host cell. That's permeability is how easily the membrane can let molecules through it, and virus host cell interactions, okay? Then there's a membrane protein here. And then there's some more proteins here which are involved with structural accessory proteins. These are just extra things. And then there's an N, which is the nucleic capsid protein. Okay, the capsid I described previously as being a shell of protein, the main shell of the virus. Inside that is this N thing, this nucleic capsid protein, which is actually bound to the RNA. The RNA is covered in it. It's coated in it, and this allows the virus to hijack the human cell, okay? Then you got, I mentioned all these things are extra genes. The spike protein itself can be, that is two sections, okay? S1 subunit and the S2 subunit, okay? The one end of this thing is a single peptide, SP. Then you got the receptor binding domain. That section binds to receptors in the host cell and the human cells. That point is a cleavage site where this thing is actually chopped in half. And then you got a fusion peptide here. That section is involved in fusing to the human cells. Then you got two heptide repeats. Heptide means it's a multiple repeats of amino acid. And then you got a transmembrane domain here. The transmembrane means a protein that goes right through the membrane. A membrane is a lipid, it's a fat layer. And the transmembrane goes right through, not on top, it doesn't sit on top, it doesn't sit underneath, it actually goes right through it. That's a transmembrane domain. And then lastly, there's a cytoplasmic domain here, which is involved with the host cell, human cell interactions, okay? The whole length of that is 1,255 amino acids, okay? The S1 subunit here, this section is involved with recognizing human cells, and this S2 subunit is involved with fusion of the viral and host cell membranes. Fusion of the viral and human cell membranes, okay? So, hope that's clear. Have a good day, and I'll see you next video. Please like, subscribe, comment, press bell, help me get to 50 subscribers. Thank you. I hope you find this interesting, by the way. It's just how the genes are laid out and the structure of the main spike protein. Okay?